Okay, now we're good. Yeah, because the big T is dominant, and so you only need one allele of a dominant to look dominant. For recessive, you need both. You have to have two recessive alleles to look recessive. Yeah. Yeah. 100% tall. What's your genotypic ratio? Well, there isn't one. It's just 1. Exactly. And then this is 1-to-1, or half-to-half, 50-50. So if you do this and you get half tall and half short, you know that unknown dominant phenotype had to be heterozygous. So that's how you can figure out what the genotype is without doing molecular biology, right? Just doing that. No. So in which I didn't realize that question was on the quiz because I didn't talk about it. Okay. So we're going to talk a little bit more about Mendelian genetics. We're only going to cover one, not exception, but one other type of inheritance aside from complete dominance. Everything I've talked about so far is complete dominance, right? If you have at least one dominant allele, you look dominant. So homozygous dominant, heterozygous dominant look phenotypically exactly the same. Homozygous recessive is the one that looks different. You will have two phenotypes, right, but three different genotypes. That's complete dominance. That's all we've talked about so far. The only exception to that we're going to do in here is blood type, just because it's fun, right, and we can play Maury-Povich paternity testing using the Punnett squares for that too. Okay, it's just another example. And so and blood typing gives us two examples of other not weird genetics things, but different things that can happen. So for the complete dominant step, the traits I've talked about so far, and all the diseases, cystic fibrosis, albinism, lobster claw, hitchhiker's thumb, all of tongue rolling, all of those have only two alleles that exist in the world, in the gene pool, okay? You're either a tongue roller or you're not. You either have attached or detached, right? You either have the albinism allele or a normal allele. For blood types, there's more than two alleles out there in the world, in the gene pool. Any one of us can only have two of them at a time. Why can we only have two alleles at a time? Why can't we have, because we're diploid, and because of our number 1 and number 1 chromosome allele, the only individuals who have 3 alleles of any gene are like a trisomy 21. Yeah, then on that, where those 3 chromosomes, they have 3 alleles. Right? But that's not where the blood type resides anyway, so it doesn't affect blood typing. Right? So even though there's more alleles out there, all of us can still only have 2 of them. But that does provide for more phenotypic variance in our population, right, than just A or B. Right? What are all the blood types that we have? Right there. Yeah, you can be A, you can be B, you can be AB, or you can be O. If we only had 2 alleles, we can't have that many phenotypes. The math doesn't work out. And so as it turns out, there are 3 alleles for the blood types. And this is just our ABO blood types. It's not the positive and the negative. We're going to do that on the next slide. But just type A, type B, type AB, and type O. So there's 3 different alleles. And because there's more than 2 alleles, they use the superscript to designate the different alleles rather than, because there's only capitals and lowercase. We don't have another kind of, cursive, to denote which ones these are. And in this case, there is a dominance and a recessive nature to these, but 2 of the alleles are dominant. They're co-dominant to each other, and they're dominant over one of the other ones. And so we can't just give them, A or B or O as our alleles. because in genetics that suggests those are different genes. Right? All the alleles always have the same letter, big B, little b, big R, little r. So if you have more than 2 alleles, you have to do something else to show that the I means it's a single gene, the subscript or the lowercase means it's a different allele. Right? Because this would get very confusing. You'd think they're multiple genes. And when we do the 2-gene cross, you'll see why you can't. Anytime there's more than one letter, it suggests there's more than one gene, not just allele. So the alleles need to have the same letters. So the capital I's are dominant, and they show them with the capital A, and little I is recessive. So type O, oh, that fell off my little slide. Type O blood is the homozygous recessive, little I, little I. A is dominant over little I or O, B is dominant over O, and A and B are co-dominant. Neither one wins. So if you have one allele you got from mom, woo, bless you, with the A allele and an allele you got from dad at the B allele, your blood type is AB. You have both. Neither one masks the other one. So they have to be co-dominant to each other, right? Because they both show up. Remember, recessive means it doesn't show up in the phenotype. Both A and B show up in the phenotype. Right? Your blood type is AB. To be type A, you can either be homozygous A or heterozygous A. Right? Big IA, or big IA, little I. Right? So if your dad is type O and your mom is type A, you could be, at least one of your alleles has to be little I, because that's all dad had to give away, and if he's really your father, you've got one. Okay? And then mom could have either given you an A or a little I, depending on if she was homozygous or heterozygous. Right? And then type B is the same way. Either homozygous or heterozygous is possible. Okay. So if we were going to do a Punnett square, which we're all going to do, and then we're going to screenshot and upload, because you know how we are. Let's just do that one. Let's do type A by type O. Okay, we'll do heterozygous A to O. You fill out your little square and you'll screenshot and upload. So this is just a standard 1-gene cross, one normal, right? There's another allele out there, but you see that each person only has, right, you can't be III or IIID, or you can only have 2. You only have that pair of homologous chromosomes to hold them. And what are all the possible blood types of the offspring of this couple? You should write the real thing just so you don't get confused, but yeah, if you just write a big capital, okay. For the uploads, it doesn't matter. But they'll be shown like, like this on the exam, so just as long as you know. So then what's this guy, sorry. So if I ask for blood type, right, for these guys, you'd say their blood type is A, right? Their genotype is heterozygous A. And what about these guys? Yeah, their blood type is O. So this would be like a test cross, right? If you didn't know if mom's just blood type A, the Red Cross just tells you blood type A. They don't tell you homozygous, heterozygous, right? And dad's O, so then that couple decides to have 10 kids, right, to make sure their numbers are good enough for probability. And if at least one of the offspring is type O, they know mom has to be heterozygous. Now, they'd have to have 100 kids to really prove it or support it. If there's at least one O, even if they have 2 kids, you know what mom is. But if, if everybody turns out to be type A, you'd have to have at least 100 kids to be sure, and then that would just be wrong. Yep, please. Whatever the first one of today's deal. Yeah. Yeah, it's implied, but it would be accurate to say it's homozygous O. Yeah. Because there is some weirdo crap out there that we're not going to talk about, that you can be type O and really have an allele for something else. That allele's mutated. It's like the Bombay phenotype or some crazy shit, but we're not going to talk about that. That's other fun, nerdy genetic stuff. Right. And then we could also play little games like, let's see, I always screw this up too. Let's say mom. Okay, so we don't know who the dad is. Okay, we're playing Who's Your Daddy. And so we know mom. Oh, well, actually, yeah, let's just know what mom is. Okay, now I know what I'm doing. Sorry, I was having a moment. Okay. Yeah, and again, which side you put stuff on, it really doesn't matter. By convention, yeah, I kind of always put it down there. I don't know why, but it doesn't matter. You can do whatever you want. Yeah. Yeah, so let's Moodle upload the one you guys just did, and then we'll Moodle upload this one too. So this would be like a question I might ask on an exam, or some fun question. Okay, so a mom who's type B has a baby so we know this baby exists. This is baby with type O blood. Okay, and she's got 5 potential fathers lined up behind the curtain. Right? And they're trying to prove that they're not the father or whatever. Okay? And so then our question is, so because the baby's type O, we know mom has to be heterozygous B. Why do we know that? Yeah, because she gave one away, so she had to have it. Right? One of this eye, we'll say, is this eye. One of the eyes had to come from mom. The other eye, has to come from dad. So the father has to have at least one little eye. So which blood types of the men behind the curtain they cannot prove they're not it. They're totally screwed if they're type, if they're type AB, they're off. Yeah, right. If they're type AB, are they off the hook? Yeah, because they can't give up a little, right? So the 2 guys behind the curtain that are type AB, do a victory dance and leave. Right? Who totally, there's no possible way they could even argue at all that they're not. Right. O for sure, right? Because O's got 2 little eyes. There's no way you could say, oh, but I'm homozygous A. Right? Anybody who's type A blood could be this or could be this. So then we'd have to do PCR sequencing or something else to get this guy off, that guy off the hook. Yeah, type B and type A are screwed too. On Maury Povich, they're not going to be doing any molecular biology. So they're still, those 3 are the candidates, right? And so they're not usually super smart there. And so they'll argue, but I'm type A, I can't have a type O baby. And then you guys would be like, no you didn't, because they could be heterozygous. And give that away. If they're O, then he can't argue at all. You have 2 little eyes, it's totally possible for you to be the dad. But the type A person might argue, but I'm type A, how could I possibly have type O? And then we explain to them that if you're heterozygous A, if you were homozygous A, then you're off the hook. But you can't tell that just from your Red Cross card of blood type, right? You'd have to do more than that. Yes. Yes. Just write something that makes sense. So the type A guy, so how could the type A guy prove or support or whatever that he's homozygous A? Are there any people out there related to him that he could bring in a blood type card and a specific blood type would totally get him off the hook? His parents. What do both his parents have to be for him to be homozygous A? But we don't know because they're just a type listed on their stupid Red Cross card. Because we only know phenotype from that. What phenotype, which tells their genotype, would they have to be for him to be A and get 2 A's? AB, right? They both have to be AB. Let's do a square, shall we? I love it. So here's our man, who's he's type A, and we want to know what his parents are that can make sure dad has to have an A and mom has to have an A. Right. Yeah, do the second upload on that one. Right? Sure, what the hell? I know, it's points, people. Right. Can you take a picture of the screen and upload it. I don't want to draw anymore, guys. You're killing me. No. But we wouldn't know just by their type. Yes, they could in reality be homozygous dominant A, homozygous dominant A. He can only be homozygous dominant A. But if all he has is their Red Cross blood type card, it just says A. It's that stupid A problem. Like, I'm type A, and I don't know if I'm homozygous or heterozygous. And you know that keeps me up at night. I can't sleep thinking about my genotype. Yeah. Yeah. So if they both have the Red Cross card that says AB, they don't have any I's to give him. He must have gotten, and he's type A, he had to have been, right? His siblings, right? He could have siblings that are AB. He could have siblings that are B. He could have siblings that are AB. But we know he's type A. Right, so, sure, we can upload it. What the hell? I think there's 3 today. If we have another, I'll just make another one. I have the power right here at Moodle to add an upload. So does this make sense? Yes? No? No, you do not. Right. Yeah, so now he does a victory dance and walks off the show. So those are the kind of questions, right? You have all the tools. You know dominant and recessive. You know the blood types and what they mean. And then I present you a situation where you have to think about all the stuff you know to come up with your answer. You might have to draw a couple squares or at least one, right? I can give you the phenotype or the genotype of the child. You tell me the parents. I can give you the parents. You tell me the child. Tell me the grandparents.
Are all dominant genes co-dominant? Can all dominant genes be co-dominant?
That's a good question. His question is, can all dominant genes be co-dominant? If there's multiple alleles, there's usually some co-dominance going on or a dominant series where this one's dominant over this one, but not over that one, which is dominant over both of them. But we're not going to do that in here. We get to those more complicated situations when we get to 360. And then the other picture I showed, there's also the possibility that two alleles are incompletely dominant to each other, which means the heterozygote looks like a completely different phenotype than either of the other two phenotypes. But we're not, yeah, so that would be like a white flower and a red flower results in pink, right? It's not red, it's not white, it's a completely different phenotype. Whereas codominance, you actually see both traits. That would be like a striped parent and a spotted parent are crossed, you get striped and spotted. It's not like completely different. It's not zigzags. That would be incomplete. If both show up, it's codominant. If something completely different shows up, they call it incomplete dominant. It's just the way those alleles interact. Oh, she's down. Yeah. No. Right, so the blending inheritance said that pink now is a new entity, and if we go pink on pink, it's always pink. And we know that's not true, right? Because if pink was the heterozygote, and we do hetero on hetero, we know we go back to having that 3-to-1 ratio of phenotypes. So that's what sort of threw blending to crap. Right? Blending suggests that this is now an entity that can never be separated. Pink will be pink, 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 pink. But they're not. They're complete particulate units, and when that white pops back out, and a purple, actually it's 1 to 2 to 1, sorry, yeah. The purple, pink, this is purple, pink, and then white comes back again. Right? So blending, but that's where that hypothesis came from, because sometimes white sheep, black sheep looks gray, but then gray on gray will give you white, gray, and black again. And there was another question? Yeah.
So the blending is not based on the genetics?
Right, that's just what people were seeing, and so they thought, oh, somehow, and especially with humans, because we have a billion different features and traits and so forth, and kids look like a blend of parents a lot of times. Right, and then their kids look like a blend of them, which looks like a blend of the parent, and so that sort of misconception was passed, like somehow the genes were combining in, or whatever it was that was causing it was changing and combining each generation, and they're not. It's just how they interact with each other. So yeah, there's a lot of other crazy stuff going on in genetics, but we're just doing the basics in here. Okay, and then Rh factor is just the positive and negative. That's a completely different gene. It's back to only 2 alleles under complete dominance. So it acts just like any other gene. It's just another protein on the surface of your blood cells. Okay, so negative is recessive. So if you're negative, right, that means your phenotype is you're negative. So I'm A negative. I don't know what my genotype is for the A. I could be homozygous A or heterozygous A, but I know I'm homozygous recessive for the Rh factor because I'm Rh negative. Whereas if you're a heterozygote, you're positive, you're Rh positive, and obviously if you're homozygote, you're Rh positive. Okay. So if you're AB positive, you know your genotype for your blood type, but you don't necessarily know what your genotype is for the Rh factor. You could be homozygous or heterozygous. But if you're AB negative, you know it for both. Right? Your genotype for your blood type is AB. Your genotype for the Rh factor is negative, negative. And so then you can throw the blood type into Maury-Povich also. Right? If somebody's if the baby is negative and mom's positive, and we don't know what dad is, right? Obviously mom has to be heterozygous in order to have a baby that's negative. And then if, dad can't really get away with it for either. If he's positive, he could be that and he could be the father. And if he's negative, he could be that and he's the father. So there's no way to get rid of them. Oh old talk show sleazeball host guy. It's like on old. I know, but he's on like at midnight or 2 a.m. on some. Oh, is it still during the day? At the gym he was on? All right, go to the gym, people. Watch some good quality TV. Yes.
The incomplete dominant and co-dominant, is it considered homozygous?
Heterozygous. Always hetero, yeah. Those two classes are always hetero. So if mom's negative and baby's positive, right, we know mom's genotype. We don't know, right, what this other allele is for this baby. And so again, right, if the dad's negative, this still could be negative. And if he's positive, that could be positive or negative. So only sort of with a combo of parents, right, this guy, the positive had to come. Oh shit! I'm fucking this up. Had to come from the dad, right? Because mom can't give a negative. Mom had to give this one. The positive had to come from dad. So if one of the guys backstage are negative, then they're off the hook because they have no positive to give away. Right? Because two parents that are both negative can only have negative offspring. So there's no way that he could be the dad because the baby is positive. It's just a protein that sits on the surface of a blood cell. It's named Rh because it's one of the proteins that causes a huge immune response when you do transfusions to other animals. When they were originally doing xenographic blood and trying to do transplants from monkeys to people or chimps to people, like if you try to give rhesus monkeys, which are one of the laboratory animals, human blood, they have a huge immune response, and it's to that specific protein. So they called it the rhesus factor, so the Rh factor. It's just a highly immunogenic molecule, and so that is one of the things, like you can't give somebody who's negative positive transfusion because you will create such a huge immune response to it that you actually have anaphylaxis and die. So when they were doing those, like why sometimes when you transfuse from you to you, it's fine and they live, and other times they have this huge anaphylactic shock and die. And so that's how they found all the different blood types and the Rh factor. There's tons of other proteins on the surface of these cells, but none of them cause that huge immune response, right? So for just getting a blood transfusion, you want to match the type and the Rh factor. So if you go to the emergency room and you're bleeding out, they're going to give you O negative because everybody can take O negative. Because O means no proteins on the surface for the type, and negative is no Rh on the surface. So anybody can get O negative. Okay? So that's the emergency room blood type. So if you're O negative, the Red Cross is constantly sending you information, please donate, please donate, please donate, because you're the universal donor for emergencies. If you go to the ER and they're like, oh, you need to have surgery, they're going to blood type you, and then they're going to, if they need to transfuse, they're going to give you A positive if you're A positive. They're not going to waste the O negative when they know what you are. But they will blood type you every single time before a procedure. Right? Let's say you go in and you have a biopsy. They blood type you before it. If you go the very next day and they do another surgery, they blood type you again. Because if they make a mistake, if the nurse wrote down the wrong type on your chart, you're dead. So it's not worth, right? So it's irritating. O negative can give to everybody. O negative can only receive O negative. Right? AB positive can receive from everybody because they've got everything. But they can't give to anybody but AB positive. Right? So they always want to give you your exact blood type when they can. if it's an emergency, they don't have time to type you, you're going to die because you're bleeding out, then you get O negative. But otherwise, if every little thing you go in for, they type you because they don't want to just trust that chart because it's so important. Yeah. Anytime blood is donated, that's all screened for all of that stuff. So unless you get a transfusion in another country where they don't screen, like the blood is very safe here, right? All the retroviruses are tested for, HIV, HTLV, all of that, and all the hepatitises. Now, before hepatitis C was even known to exist, yeah, some people had it and they gave it to other people and transfused 20, 30 years ago, right? Same with HIV. When we didn't know it existed before 1980, how could you test for it? So, it's within the realm of possibility some emerging viruses out there that if you get a blood transfusion, you're going to get that. But everything that we know about is screened for. So if you're having a planned surgery, you give your own blood, and they stock it for you in the case of emergency. You're just getting your appendix out or just something, non-emergency. You give blood the week before. They store it just for you. You have your surgery. If you need it, they give you your own blood. Right? If you don't need it, then it donates into the pool. They tested it for everything, blah blah blah, and then it'll just be donated. So it's safest, to give your own, or you can have friends come and donate just for you. You're going to need a transfusion during the surgery. You can have people come and that's your blood type come and donate. They screen it all, and then that's what they save for you. Okay. Oh my God, we're behind. So we did a lot of uploading. So that's good. Okay, so I posted that little sex determination video, so you guys can watch that. And it's essentially just, right, we talked about it before, eggs are only X's, sperms are X and Y. So, Dad, it's your fault if you have too many of one type and none of the other. Too many of any type. Yeah. No, so X and Y are both considered the sex chromosomes. The autosomes are 1 through 22, anything that's a non-sex chromosome, right? But there's all kinds of terms for, right, because the X has its own pair, right, it has a homologous chromosome because in a female, 2 X's. If you're X and Y, you're considered hemizygous. You're not really heterozygous. It doesn't really exist, but it has this other name. If you're boys, you're also heterogametic because you have 2 different sex chromosomes, whereas females have the same. Okay, so practice the practice problems for the 1-gene traits. I'll post the key later if I can remember to do that today. Or somebody email me if it doesn't pop into Moodle, in case you need to check your answers. If you want more practice, all you have to do is Google monohybrid genetics or Mendelian genetics quiz or Mendelian genetics problems, and you'll get a cajillion different sites where you can go either take quizzes or do problems with answers, find worksheets, all kinds of crap if you want to practice, like the stuff that was on the quiz today. Right? If those all gave you a hard time and I'm not letting you see them, there's a billion questions out there that might look very familiar to you. Where do you think I get my questions from? For quizzes, right, that are out there. And if you don't get it, then you can print those, come into office hours, we can work through it or something like that. Okay, so there's lots of resources out there. I'm not trying to hide so you don't get a lot of practice. I just need to be able to sort of recycle the same questions because it's a lot of work just typing in questions and all that stuff for quizzes. Not to be mean. Yeah.
On the test, some of these test questions are just how we were doing it earlier, where there's plenty of that was, and the only way you can get out of it was
Yeah, there's likely to be one question about paternity and blood typing.
Would that include grandparents, like to get out of it?
No, I won't be that hard. I would do that to my 360 people. It just popped into my head and that just made me excited. But yeah, I won't do that for you guys. So it'll be like you'll know mom and baby and something about dad, or, yeah. Is that cool? You guys are cool with those? And same with these 2 gene trait ones. You can search that online as well for extra practice. There should be a practice sheet posted for today for these times. Some are a lot harder than they need to be because I just pulled them from genetics textbooks. But if you can work through them and understand it, looking at your notes with help, then you'll be fine on the really hard ones. And they're all essentially based on the same monohybrid rules that we've already learned. Okay? It's just we're talking about 2 genes at the same time instead of doing them separately. If you do a 2-gene cross separately, you're going to get the wrong answers because they're dependent on each other and who gets what. Okay? So I'll go through these and then we'll do at least a couple examples, I think. And so when we're talking about, and how do you know if I'm talking about 2 traits or 1 trait? You're actually physically, I'm telling you the phenotype about 2 different things. Right? Tall and short is 1 phenotype. 2 alleles, 2 types of the same gene, height. But if I'm talking about the plant is tall and green, or tall and yellow, or wrinkled and stinky, I don't know, right? That's 2 different things. So, in the paragraph listing what we're talking about, if it's giving you 2 things, you ought to, 2 genes. I need to keep track of these. Some of them will be easy, and I'll tell you the genotypes. You'll get the phenotypes and the genotypes. You just have to do the cross. The other ones, you'll get phenotype. You have to figure out the genotype of at least one of them, or, heterozygous at both traits. What does that mean? Big T, big R, little r, right? Heterozygous, homozygous for tall, heterozygous for color. Homozygous would be big T, heterozygous for color, big G, little g. Right? To be able to think about phenotypes, what is their genotype? I give you the genotype, you tell me the phenotype. Okay? And so it looks, right, something like that. And so color and shape are 2 totally different traits. So color, we're using 1 alphabet letter, capital for dominant, lowercase for recessive, just like we did for the 1 gene traits. The other gene, a different letter, big R for the dominant, little r for the recessive. So we know because we're using 2 different letters that it's 2 different genes. If it were just multiple alleles, right, we'd have Y superscript A, Y superscript B, something like that, if it was just multiple alleles. The only multiple allele one you guys are going to get is blood type, so you don't have to worry about, that sort of thing. You'll recognize the blood type questions, those have multiple alleles. Everything else we do is just going to be 2 alleles. So 2 alleles for each, 2 genes. So if they're heterozygous for both traits, right, they're going to look dominant for both traits, right? Because the heterozygote's under complete dominance, which all these are going to be under that umbrella again. As long as you have 1 capital letter of that gene, it looks dominant. And you'll either be told which one's dominant or which one's recessive, or you have to figure it out. How did we figure out which one was dominant and which one was recessive for a 1-gene cross? Do you remember what that setup had to be? A test cross, exactly, to look at which one of 1 parent phenotype, the dominant phenotype, is heterozygous or homozygous. But if we're just given, remember, purple and white, yeah, we want to look at the F1. We set up the parentals, right? We make the 2 true breedings, and then whatever shows up phenotypically in the F1, the heterozygote, that's the dominant phenotype. So it's the same for a 2-gene cross, except we're just looking at both, what am I looking at? Color and shape, right? So we'd set up the the parental, which has to be true breeding, right? And so, right, one parent could be true breeding dominant, the other could be true breeding recessive. In this case, it really doesn't matter if one parent is dominant for one and recessive for the other. They don't have to both be dominant and both be recessive to be considered parental. Because if you cross these two or these two, you end up getting the same ratios. They'll be in different squares in your Punnett square, but when you count them up, it'll be the same. It'll end up looking the same. All of these guys are going to be heterozygous. They're all going to end up big R, little r, big Y, little y. Like, that's all they can be. That's all the parents have to give away. Okay. By convention, we usually show them like this, but not to confuse you that it can't look like that. Right? As long as they're opposite, the parents, this big R's, little r's, big Y's, little y's. You can't have, if there's big R's over here, you can't also have big R's over here, because then that's not the parental cross, because you want dominant recessive. Okay. So to make our Punnett squares, this is where sometimes people get a little crazy. Okay. It's the same idea as for the monohybrid, but we have to deal with two different genes and the combinations of gametes that could be made from that. So, let's squeeze this up here a little bit. Whoa. Okay, so this guy down here is saying, okay, that if we cross either of these parentals, we get the heterozygote, right? So they're showing us here. All the offspring in the S1 are heterozygous. And because they already told us which ones are dominant and recessive, we know this guy is yellow and round. Right, because we told you that. But whatever that guy's phenotype happened to turn out is whatever's dominant. So if we weren't told, whatever that guy looks like, now we know which is the dominant trait. And then if we want to do this cross to get to that F2, we have to figure out, well, how do we set up our square? What are the gametes? Right? When we just had this guy, we knew half the gametes were going to be big R and half were going to be little r. And then that's, we just put those, and then the other one, right? And that's how we set up our square. Really, the things on the side are the gametes, things on the top are the gametes, and then the square helps us do all the different possibilities. Right? That's all the stupid squares for. When we have these two, we have to consider both of those genes and all the different combinations of gametes. And so this is saying, here, if we have this big Y and this big R could get together into a gamete. Right? If we're thinking about meiosis, they're on two different chromosomes. One goes this way, one goes that way, or the other one can go this way. Right? Or this Y could have been on the chromosome that gets stuck with that guy. So this one and this one could have gotten together in one of the gametes. This guy and this guy could have gotten together, this guy and this guy. If you need to set up meiosis using these two different homologous chromosome sets and do that, and have them labeled on the chromosomes, do that, so that you understand how all four of these gametes could form. Okay? Because that's just independent assortment, that's just meiosis. So all the combinations could happen in this one individual to make the gametes. This is just making gametes. We haven't started talking about making the offspring yet. Like, that's just what we line up on our Punnett square. And then the other parent obviously is the same genotype because it's F1 on F1. And so the other parent would make these exact things. And you'd end up with a square with 16 squares in it, which is a lot of squares. And so that's what they show us here, right? Here's all the gametes. Big R, big Y, big R, little y, little r, little y, little r, big Y. All the possibilities, those four, lined up on a Punnett square. okay? And then filled in for you, because that's going to take me way too long. It'll take me all classes to fill that out. Okay. But again, it's over and down, right? One big R from this guy, one big R here, means that guy's big R, big Y, big Y, big Y, big Y, big Y. So this guy is homozygous dominant for both traits, right? He looks round and yellow. And you can follow all of these down and across. So you build the square just like you do with two gametes each, but there's 4 possible gametes. Okay. This, and again, just like the monohybrid, no matter what 2 heterozygotes with a 2-gene cross you put in there, you will always get the same ratios in the square, right? Remember for the 1-gene square, it was 3-to-1. You got 1 homozygous dominant, 2 heterozygotes, and a homozygous recessive from big R, little r, big R, little r. A 3-to-1 phenotypic ratio, 1 to 2 to 1 genotypic ratio. It's the same thing here, except we don't worry about the genotypic ratio because there's too many of them, so we don't care. Okay? We care about the phenotypic ratios. And the phenotypic ratios then tells us the genotypic ratios. So what the hell do I mean by that? If we go through here and we count all of the double dominants, so look at your guy and tell me how many in this big square are both round and yellow. You bet. So everybody count just so it makes sense to you. All right, so 9. So there's 9 that we know have at least 1 big R, right? To look dominant, you have to have at least 1 big R. To look round, yeah. And at least 1 big Y to be yellow. So the phenotype is round and yellow, and there's 9 of them. 9 out of how many is in this square? Yeah, 9 out of 16. And we'll just keep that in mind. So though all the possible combinations, if you look at all these, big R works, big Y works. Big R, big Y, little r, big R, big Y, all the different possible combinations, as long as we have at least 1 big R and 1 big Y, we know they will look round and yellow. They have to. Okay? All right, now let's count up 1 trait that's dominant and 1 that's recessive. So let's go with green is the recessive and round is the dominant. Yeah. Okay, that's so good. And so what does that guy have to be? What did I said green, right? And round. So the round has to be, exactly, capital R. Yeah. So look, oh shoot, that's not the right letters. I made G's for green. Right, does that make sense? Anything little y is going to look green, because that's recessive. And what about the next category? What about the opposite? What about, yeah, wrinkled and yellow, you're old wrinkly. Right, that's the genotype. So that one's easy. It's either little r, big Y, or little r, big Y, little y. Right, there's only 2 options for these guys, but there's a whole crap load of different options for the 9. And then what's the lonely little one? He's the big loser? Yeah, the double recessive. Little r, little y, little y. So the phenotypic ratio is always 9 to 3 to 3 to 1. We know what the 9 means, right? At least 1 big R, 1 big Y, at least 1 dominant of each trait. We know both 3 categories have to have 1 dominant, 1 recessive, the other dominant, the other recessive, and the 1 is always homozygous recessive. So that one's easy. The phenotype, there's only 1 of them, and there's only 1 genotype of that guy. Okay? So you should never, ever have to draw out this square, especially on a test, because it's going to take a long time to draw 16 squares and fill all that out, and then count up when I ask you how many are going to be dominant for one trait and recessive for the other. All you need to do is remember this and go 3 out of 16 are going to be dominant for one and recessive for the other. And then another 3 is recessive for one, dominant for the other. Dominant, dominant, 9 out of 16. Recessive, recessive, 1 out of 16. Right? That will never change. It's like big G, little t. Doesn't matter. Big R, little r, big Q, little q. Heterozygous on heterozygous. And F1 cross onto F1 to get to the F2 is always, always, always this. Always 9 to 3 to 3 to 1. Okay. You can draw it out at home just to make sure you trust yourself, right? And you believe me that this is true, but I swear it's true. Okay. So I'm never going to I'm going to ask you questions about that, but they're going to be questions about 9 to 3 to 3 to 1 and which genotypes fit in which categories and that kind of stuff, right? So that honestly is a memorizing thing, right? Like I said, memorizing is not bad, it's just not all we should do, right? But that's something we need to memorize and then understand it, why those numbers are what they are, okay? But for most 2-gene crosses, especially ones I'm going to ask you something about how many offspring do whatever, as long as one of them is homozygous, your square is never going to be 16 squares because some of the lines will be redundant, right? And that goes back to when we talked about, remember when I was making fun of them because they only put one R in one and they just did that. And I was like, that's confusing because what happened to all the other gametes? Well, they really do exist, right? You can put both, but you're still going to get the same ratio. They're still 100% big R, little r. Okay? And so that's the same with the 2-gene. If there's anything redundant, you don't have to list it as a square because it's a ratio, so it's not going to change 50-50 or 75-25. So what do I mean by that? I mean, if you have this kind of cross, right, big T, little t, then this guy and that guy, which one goes where? This one. So, here, let me get rid of that because that's messy. So this parent is up here. His possible offspring are big T, big P, or big T, little p, right? So isn't that all the gametes that guy could make? But he'll make 2 big T, big P's and 2 big T, little p's. Okay, but you don't have to list out that extra big, right? We don't need to put that one there. Or this one, because it's exactly the same as the one's already listed. We could, but we don't have to. And same with this one, right? Big T, little t, little t, little t. Well, big T with the other little t with the other little t. Right? We could add that, and now guess what? We're back to our 16 squares. But the ratio is going to end up being the same if we just do these as if we do all of them. So we're not going to waste 5 more minutes on an exam filling in a bunch of squares that don't matter. Okay? So if you have a redundant gamete, we're already showing that one. We don't need to show this one. Half are big T, half are big T, little t. So we don't need to put those guys in there. Same with this one. Half are big T, little p, half are little t, little t. We don't have to show them because this still represents half and half. Half and half. So if you fill that out, right, this is what you get in the squares. And we would say, how many different genotypes are in this? 4, right? All are different genotypes. So what about phenotypes? We don't really know what they stand for, but as far as recessive and dominant. Yeah, 2, right? This is double dominant. This is double dominant. This is dominant recessive. Oops, I guess I could do a different color. And this is dominant recessive. So genotypes, we'd say 1 to 1 to 1 to 1. Phenotypes is just 1 to 1, right? Or 1/4, 1/4, 1/4, 1/4, 1/2, to 1/2. What's that? Okay, then I just wrote that out for you. See how nice I am?